Ja, då går vi vidare i programmet och nu så kommer vi att få lyssna till ett bolag som för många är känt. Känt utifrån det perspektivet att ni har träffat dem, lyssnat på dem. Bolaget har också varit listat på aktietorget under ett antal år. Och jag vet att det här ute bland åhörarna finns många följare. Jag vet också att det finns många följare bland er som sitter och lyssnar på denna webbsändning. Jag vet också att när WntResearch, som vi ska lyssna till, var och presenterade sig på Sedermeradagen nere i Lund, så var det en lång räcka utav frågor som besvarades utav Nils Brunner. Välkommen tillbaka, Nils.
Tack så mycket. Kan jag få den på här också, presentationen?
Jag ska se vad jag kan göra. Börja det.
Jag heter Nils Brunner. Jag är född i Borås. Så jag har varit i Göteborg många gånger. När jag blandar dansk och svensk och ger presentation, så någon gång så funkar det inte här inne. Så därför ger jag min presentation på engelska. Jag hoppas det är okej. Så
jag ska se vad jag kan göra.
Du ska se vad okay. So what I would like today to do is to give you an update on WntResearch and tell you where we are in our development and what will come in the near future. And WntResearch is a company that is developing anti-cancer drugs. See if this works. Yeah. And why are we doing this? Why is it so important? So if I ask each of you to raise your hand if you had somebody in your near family or some good friends who have cancer, you would all raise your hand. This is a disease that is almost epidemic. So if we live long enough, we will all get cancer, unfortunately. These are the numbers from Sweden, and you can see that there's a lot of Swedes, like Danes, like Norwegians, like in any country, who gets cancer. But what is even worse is the number here that there are so many people dying from cancer. And they die from not the primary tumor, but they die from the spreading, as we just heard the former presenter, the spreading of cancer. That is what kills the cancer patients. So we have a mission, for sure. We need to combat cancer. We need to eradicate that disease if possible. So I have divided my presentation. I'm sorry that I can't see my presentation over here. I dislike to talk to the screen here, but that's probably how it's going to be. So we'll talk a little bit about WntResearch background. I'll give you a little lesson on cancer. I'm a professor at the University of Copenhagen teaching tumor biology. I'll tell you a little bit about our lead compound, FOXY5. Then we'll go directly to our ongoing phase 1 study. I'll tell you something about the next study, which is a phase 1b study, and why we have a phase 1b study. And then I will tell you how we have planned our phase 2 study. The phase 2 study is where we really want to see if it works. And then we have a second drug in development, BOX5. I'll give you a few words about that. And then finally, there will be open for questions. Okay, so I think I know Peter doesn't like that I step down here, Peter, but I need to see the screen, and if it's not here, I need to be down there. Where's Peter? Peter? I step down here because I need to see my slides here. So this is something about WntResearch. So it was founded in 2007 by Professor Tommy Anderson from Lund University in Malmö. And he did a discovery working with white blood cells, and he thought perhaps this discovery could be used in the treatment of cancer. And it had something to do with cell movement, how cells can move. And I guess you can imagine that when a cancer cell needs to spread to other organs in the body, it has to move. So he actually found a mechanism by which cells move, and he could transfer it into cancer cells. And when he blocked this mechanism, he could see that the cancer cells couldn't move any longer. So they then introduced a company on Aktietorvet in 2010 to raise money to continue the work. And we have patents on all the inventions, and almost all of the patents are now issued. We are filing new patents on the way also. We have an experienced and dedicated management team. I'll come back to that. We have a lean and focused organization, so we are saving money by all of us being part-time employed. And we have raised money a couple of times, and we also got a Eurostar grant. This is a grant you can get from EU, and we got comparable to 48 million Swedish kroner, partly from EU and partly from our partners. So we were well equipped to get started, and if I look back on these years where we have been working and where I have been a CEO, I think we have been very successful. It has been a very nice path from preclinical testing to the clinical studies, and now finalizing the phase 1, continuing. I think we couldn't have done it better than we have done. Okay, a little bit about the management team. Myself on the top here, being a professor at the University in Copenhagen. I'm also a medical oncologist, so I have a dual education, also being a molecular biologist, and have experience beforehand also from biotech. Then we have Thomas Feldhus, and we have just announced that Thomas Feldhus, who you will meet later with the company Saniona, he decided that he wants to devote his full time to his new company Saniona. So we are looking for a new CFO now. Thomas was employed 1 day a week, taking care of all the finances and business development, did an extremely good job, but I think it's natural that he now wants to dedicate his full time to his new company. Tommy Andersen, the inventor, the founder, a very talented Swedish professor in Malmö, and he is our scientific officer, and still in his laboratories doing a lot of research and also directing research that we have done other places in Europe. And then finally Ulla Bull, who is our responsible person for all our clinical trials and also for investor relationships. Okay, so a little bit about cancer. What is it? Why is it so difficult to treat cancer? What is the problem here? Why haven't we got rid of this disease many years ago? The problem is that it's a disease in the DNA. And you all know what DNA is. This is how we inherit from our parents through the DNA. This is the backbone of everything. It's a disease of the DNA. But the problem here is that it's changing over time. So what is very specific about cancer cells when you compare them to normal cells is that they change the genes. They either change the genes by having changes in the structure of the genes, or they change the expression of the genes, making it very difficult to treat them. And you know it from bacteria. We are talking about resistant bacteria. It's the same mechanism that bacteria can change according to the environment, and suddenly they are resistant to antibiotics. It's exactly the same with the cancer cells. And that's a big challenge in treating cancer. And that's why we haven't been that successful yet. Okay, so a little bit about cancer. I think the most important message to you all is that we expect, and that means WHO expects, that within the next 10 or 15 years we'll have an increase in cancer incidence between 30 and 50% because we live longer. And it's a disease that is related to age. So if we don't find new ways to treat cancer, it will totally dominate the health budgets, and there'll be so many more cancer cases, and people will die from cancer. So it's a huge unmet need here. Also, that we have, at present, we have surgery, radiation therapy, chemotherapy, targeted therapies, anti-angiogenesis therapies, and so on. But we don't have treatment that specifically target the spreading of cancer. And that's where WntResearch has a role here. Because our drug is a drug that specifically inhibits the spreading of cancer. So our drug is not killing the cancer cells. Our drug is going to be given together with chemotherapy that kills cancer cells. But those cancer cells that are not being killed, and because they're resistant, we can prevent them from spreading. This is the whole concept. Yeah. Okay, so here's another cartoon, and that just shows a little bit about the complexity of cancer. So we know that cancer cells are characterized by they grow faster, they can grow forever. Normal cells, they have a certain number of cell divisions and then they stop. But cancer cells, they can divide forever. They can prevent dying. Apoptosis means dying. They can make sure that they get a lot of nutrition by making sure that a lot of new vessels are formed around and within the tumor. They can, as we talked about, form metastases, the spreading of cancer, and they can take advantage of the environment for their growth also. And we have today, as I said, treatment that stops the proliferation and kills the cells. This is mainly chemotherapy, as we know chemotherapy. We have drugs that induce cell death. We have drug that inhibits angiogenesis, but we don't have any drugs now that can prevent the spreading of cancer, and this is our FOXY5, where it has a role. So how do we see this drug develop in the future? So it's a drug where we know about the mechanism. We know about how it works in the cells. We know the molecular mechanism of this, which is very important. We know also that patients with breast, colon, or prostate cancer will be candidates for our drug, and I'll come back showing you why we know this. It's called a peptide, a small drug, and what it's doing is that it's reconstituting a specific signaling pathway in cancer cells. So cancer cells can downregulate a mechanism that prevents them from spreading. So they can do everything. Cancer cells are very clever, and they are cheating us all the time. So they just downregulate what they don't like, and they don't like to be sitting in the same place. They want to spread. They downregulate these mechanisms that prevent them from spreading, but we reconstitute it by giving our drug, and I'll show it to you. Market is huge, as you can understand, that there are so many cancer cases, and there'll be even more. We are in the clinical trials already, and hopefully launch will be in 2019. We have good patent protection and can be even prolonged to 2028. And we are completing our phase 1 study now here, and we will start phase 1b, and as soon as possible after that, we'll start the phase 2 study. So we have, I think, a good track here, and we are in a hurry because we want to get out to the patients and give them a chance to benefit from the treatment. So a little bit about this WNT5A protein, and now we'll be a little bit technical. So as I said, this is a protein that is produced by cells, also normal cells, because normal cells need to sit there where they have their function. We don't want the intestine cells to get into the blood and go to other places. So normal cells, they sit where they're supposed to sit and do what they're supposed to do. While cancer cells, they can move around. And here is a little cartoon showing this protein that is produced by almost all cells, and it's sent out of the cells, and then it binds to another protein on the surface of cells, and then it induces a lot of different mechanisms that make sure that the cells are sitting where they're supposed to sit. But cancer cells, they downregulate it, so they can move. This is just to show you how complex it is, and due to all the research by Tommy Anderson, we know all this now. And this is very important in drug development, that you know the mechanism of action, because then we can do a lot of things. And I might say a little bit about it a little bit later. But this, I'm not going to go into details here, but it's something about cells sit together and adhere to each other, cells adhere to the environment. They can make small arms, legs out so they can move. And by reconstituting the Wnt5a pathway, we prevent all this, so the cancer cells are just sitting there. I think, yeah, okay, it works good. Okay, so here's a little cartoon just showing how it works. So in the middle you have a cancer cell, and here we have the Wnt5a protein that is secreted, as I said, binding to the surface, and then inducing cell adhesion to the outside here, here to other cells, and to the outside of the cells, the stroma, as it calls, that is out here, the connective tissue. Then FOXY5 can do the same as Wnt5a. It's a mimic. It's taken out of Wnt5a. Wnt5a is that long, FOXY5 is that long, but it can do the same. So here we give FOXY5. Oops. Yeah, okay. It doesn't work. It's supposed to show you that when we remove Wnt5a and FOXY5, the cancer cell disappears. When we reconstitute it, it's sitting there. But it's apparently hard to get this to work when you give a presentation. So I'll just continue here. So why didn't we just take Wnt5a, the natural protein that makes sure that cells are in the place where they're supposed to do? Because Wnt5a by itself is not a good drug candidate. We have to change it. It sticks to surfaces and so on, so it's hard to get it to the cancer cells. And that's why we made FOXY5. And FOXY5 can do exactly the same as Wnt5a, but it's much more easy to get out to the cancer cells. Also, Tommy Anderson invented a way to link a little extra part to FOXY5, preventing it from being degraded. And it's something that he also learned from working with white blood cells and bacteria, that now it's much more stable by putting this. And this is part, of course, of the inventive step, part of the patent protection. I said before, we know that our target population is at least breast, prostate, and colon cancer. And we know this because Tommy Anderson did some very excellent studies in clinical material also. He did take a large number of cancer patients, got access to their tumor tissue, here, yeah, and then he tested whether there was a difference in survival in metastatic spread, whether you had Wnt5a or not Wnt5a. And I'll show you these pictures. And also he did some very nice animal models where he injected cancer cells into the animals, knowing that if he didn't do anything, the cancer cells would spread, for example, from the mammary gland, where he injected breast cancer cells, to the liver and to the lungs. But if he added FOXY5, he prevented the spreading of the cancer. So I'll speed up a little bit here, I think. This is just these data from the clinical studies Tommy Anderson did. This is in breast cancer, this is in colon cancer. Here is time from surgery. So the patients were followed for a long time, and this is recurrence-free survival. So in the beginning, nobody had got a recurrence, but over time they get a recurrence. If they have Wnt5a, the cancer cells cannot move, and they do not recur, they don't get metastasis, while if they don't, they do here. It's the same for overall survival in colon cancer, and I could have shown you a similar picture in prostate cancer that Tommy Anderson has also published. It's the same feature. Lack of Wnt5a, it goes really bad, they get relapse and they die from the disease, and that's why we reconstitute Wnt5a. Okay, this is just a little bit about one of these preclinical models, and I think I'll go to the next slide already because this is some text. I'll just show you the cartoon here, and this is important. So FOXY5 does not inhibit the growth, as I said. So we need to combine FOXY5 with chemotherapy. As you see here, this is in mice with tumors, that the volume, this means the growth, there's no difference whether they get FOXY5 or just saline, and the animals, there's no toxicity, but here is what is very important. See here, if you give the animals FOXY5, and these are lung spread. There's hardly any lung metastasis or liver metastasis, while in the 2 control groups, the cancer cells spread. So this is what we call preclinical proof of concept. And Tommy Anderson, as some of you also know, they have just presented new data at the recent American Association for Cancer Research meeting, showing that it also holds true in prostate cancer. If you inject prostate cancer cells into the prostate of mouse prostata, they spread, but if you give FOXY5, they don't spread. Okay. The message of these preclinical tox studies, and this is something everybody has to do if they want to introduce a drug into patients or human beings, they need to make sure that it's not toxic. And if it's toxic, they need to know which dose they should give. We have done it short term and are almost done with the long term. The simple message is no toxicity. Very important to us because it's got to be much more easy to give it together with chemotherapy that is toxic. We don't have to consider the toxicity. We apparently have a drug here that works beautifully in the preclinical models and hopefully in our clinical studies without being toxic. And this is also important because it's probably a long-lasting treatment in order for a long period of time to make sure that there's no cancer cells that can spread. So I'll just go over this. I can see time is running. So the phase 1 study that is running, and we just dosed number 4 out of 6 patients yesterday in the last dose. So very shortly, we have finalized the phase 1 study. There will be a report that we have already started writing because we can put all the doses from 1 to 7 together now and then just adding the last dose level. There will be a full report sent to the authorities and being in the company, but we'll make a summary that we will release on our homepage, and there will be press releases as soon as this is done. But it's just about to be done. We have been through all these steps. We are looking for biomarkers also. If you have a drug that is not toxic, you want also to have a readout for biology. So we're doing a lot of biological studies on the patients when they get the treatment to make sure that we have the right level and we see the right biological effects. We can also pass this one. I think it's important just to say a few words about the phase 1b. So we decided to introduce a phase 1b between the phase 1, ongoing phase 1, and the upcoming phase 2 study in order to learn more, to make sure that when we start the phase 2 study, we know what we are doing. We are doing the right stuff. We are doing the right test on the patients. We get the right readout. So we need to get more information. Remember that in the phase 1, dose 1, dose 2, dose 3, up to 8, we have in the first 7 doses only 3 patients at each dose level. We have interesting data, but it's all too few observations. So now we need to include some more patients to get more information, and this is a phase 1b. And personally, I expect the phase 1b to be study to be rather short term, to get the patients in and do our experiments, and then we can continue hopefully already late fall to the phase 2 study. So the phase 2 is important. The phase 2 is a study where WntResearch is going to prove the value of FOXY5. Also remember, in the phase 1 study and phase 1 study, we are only allowed to include patients who already have metastatic disease. This is the rules. It's patients where the oncologists have said, unfortunately, we don't have more treatment options for you, except you can participate in a phase 1 study, experimental, we don't know whether it works. So having an anti-metastatic drug, it's not the right patient population. What we need in the phase 2 study is to get access and include patients with no or very limited metastatic burden, and then show that FOXY5 prevents or reduces the number of patients who get metastatic disease. So these are some of the considerations that we have, and we have discussed it very thoroughly in the management and with the board and with external advisors. How do we test an anti-metastatic drug in a phase 2 setting where we could expect to get a readout? So this is how it's going to be. And if you read from here, it's patients with colon cancer or probably colorectal cancer. Stage 3 means that when they had their surgery, they had lymph nodes removed in the stomach around the tumor, and some of these lymph nodes had tumor cells, but they are removed. We know that about 50% of these patients will, within the next 2 or 3 years, get metastatic disease. They have very low burden, or you can't see any cancer cells in these patients, and we'll add FOXY5 together with chemotherapy in a randomized fashion. The other group will just have the chemotherapy. And the readout will be that those patients who were lucky to get FOXY5 will have a much less incidence of metastasis compared to the other drug. This is the closest we can get to what we can see in the animal models and how we believe FOXY5 is working. And if this works, I think we are done in WntResearch regarding FOXY5, because the exit is there. Okay, just a little note on this. I presented once before. You also have sometimes you can also be lucky, and we were lucky. A publication came out showing from another group, an American group, that Wnt5A is also involved in cancer stem cells. Cancer stem cells are the mothers of the cancer cells. They're producing the cancer cells, but they're very resistant to treatment, but they're sensitive to changes in Wnt5A. Very, very, very important for WntResearch. And we are doing a lot of stuff now on this, because this is probably the key to the resistance, to the failure. The key is to target the stem cells. Just one word, FOX5 is an antagonist. So when FOXY5 is an agonist, it reconstitutes Wnt5A. There are some cancer types like malignant melanoma where it's opposite. So Tommy Anderson also developed something that blocks Wnt5A, and we have this in preclinical trials. And my hope is that during the next 6 or 9 months, the management and the board of WntResearch will be able to take the decision to bring it further on to clinical trials, so we have our second drug candidate. Okay, why invest in WntResearch? I think there's a lot of good reasons. They're stated up here. I think we have proved until now that we can do what we promise. I think we have an excellent product here with a huge medical need. Thank you for your attention.
Tack så mycket, Nils. Då ska vi se vem som kan tänkas ha någon fråga. Ja, varsågod. Så repetera frågan. Ja.
På svenska.
Målet med en fas 1-studie är väl att egentligen se om det är toxicitet. Men ni har inkluderat också mätning av biomarkördata och så vidare. När du i tidigare kommunikationer har sagt att du har sett no negative data at all, inkluderar du bara själva huvudsyftet med toxicitet eller även gäller de övriga mätningarna?
Okay, the question is, when we have communicated that we haven't seen any effects or no negative data on the ongoing phase 1 study, is it only related to toxicity, or is it only also related to biomarker studies? It's related to toxicity and related to whatever you can consider being negative data. I'm not in position of any data that are negative regarding what we are doing. So no toxicity is excellent, but it's also creating a problem when you're doing a phase 1 study, because when do we have the right dose? Normally you would say, now we have toxicity, we can't give any more. Here is where we stop and we'll go to the next phase 1B or phase 2. We cannot do that. That's why we are so eager to do the biomarker studies and get the readout. And we are extending them even now to get more information on this. But the no negative data has nothing to do with the biomarkers. That's the toxicity. Yeah.
Okej. Tack för det svaret. Ja, varsågod.
Lite grann om era framtida kapitalbehov.
Ja.
Du nämner lite slutet här om eventuella intäkter, men hur ser det ut och när kommer nästa nyemission och så vidare?
Yeah, so the question is, if I translate it, do you expect to raise more money?
Basically.
Yeah, okay. So when we have money in the bank, fortunately, thanks to all of you who have invested in WntResearch, we have quite a lot of money in the bank. That will cover the phase 1B and hopefully most of the phase 2 or all the phase 2 study, depending on how we design the final phase 2 study. For those of you who knows a little bit about statistics, the more patients you include in a study, the smaller effects you can detect. So if we take a very limited phase 2 study, we can only see that difference between those who get FOXY5 and those who do not get FOXY5. If we increase the number of patients, we can see smaller effects. So this is a decision we have to take when we are going to start the phase 2. Will we need of more patients? Will we need more money in order to increase the number, or will we go with the money we have and the number, which is about 100 in each group? Yeah.
Okej. Ja, Alexis. Vi har ett ögonblick så ska du få en mikrofon. Varsågod.
Hej Nils. Jag skulle ställa en fråga angående den här, eftersom ni testar nu i fas 2B här, ganska svårt sjuka som har utbredda metastaseringssituation. Känner du någon oro att ni inte ska hitta någonting på de här biomarkörerna då, eller data?
Okay, so the question is, now when you are only allowed to test patients with metastatic disease, aren't you afraid that you will not see anything in your biomarker studies? I'll say no, because due to all the research by Tommy Anderson, we know the mechanism and so on. So it's actually a rather easy task to go in and see the biological effects. And now we are taking biopsies from the tumors before and during treatment, and this is the way to go. And remember also the biomarker studies that we are running now is just to make sure that we have the right concentration to the phase 2 study. So no, I'm not afraid of that, but it's a good question.
Tack för det svaret. Ja, Alexis, vi har en fråga här. Ett ögonblick så ska du få en mikrofon.
Du skriver själv här på den sista sliden då om en lättsam exit inom 3-4 år. Hur ser den ut enligt ditt drömscenario?
Det sista var i ett...
Hur ser den ut i ditt drömscenario? Den här exiten.
Mitt grundscenario.
Nej. Your biggest dream, the best.
Okay, the best dream.
The best exit ever.
Okay, so I was asked in Lund, would you like a company to buy everything here, FOXY5 and BOX5? And to be honest, no, I don't want this, because I want to be part. When we have done FOXY5, I want to be part of BOX5. I really like this part where we develop the drugs to a level where we prove that it works, and then we can have an exit. Regarding FOXY5, and this is important, personally, I would like to bring it through phase 2 because I create a lot more value for the company, and then we want an exit. On the other hand, I would also like, and that's what we're working on, to get a partner in, a clever partner as we call it, a partner, a big company that say, perhaps you want an option, this is where, or you can make other agreements, but we'll be next to you and helping you and discussing with you so you do the right things. And if you're doing that, we are interested. So there are different ways you can do it, but I would not like to sell the company today. I want to see it develop and create all the value.
Tack för det svaret. Vi har en fråga här också. Ett ögonblick så du får en mikrofon.
Jag fick delvis svar på det redan. Det handlade om stämmer att ni sökte en partner för fas 2-studien, och i så fall är det för att utöka fas 2 på vilket sätt?
Yeah, so the question here is, if you are finding a partner, could it be that this partner could help you perhaps running more phase 2 studies? Definitely, if somebody came and say, okay, we want to add a certain number of million Swedish kronor so you can run not only in colon cancer, but also in prostate or also in breast. This is something we're looking at. Definitely a possibility for us to get this and a good option.
Okej. Tack så mycket Nils för en mycket intressant presentation och för bra svar på frågorna. Nils kommer att vara kvar här ett tag till, så efter nästa presentation så finns det möjlighet att ställa ytterligare ett par frågor. Tack så mycket.
Tack.